I'd like to introduce our speaker for today, Kinjal Desai. And so Kinjal received her Bachelor of Science from St. Xavier's College in India and her Doctorate of Philosophy from Dartmouth College, USA. She is now a senior postdoctoral research fellow at the Hospital for Sick Children in Toronto as part of R.I.O.T, the research outreach team. I believe. Kinjal, feel free to correct me if I said the name wrong. But she is also working on developing a novel treatment for medulloblastoma, the most commonly occurring malignant brain tumor in children. So she is also an instructor at the University of Toronto School of Continuing Studies and has guest lectured at the University of Toronto Molecular Genetics Department. And so with that being said, I'd like to pass it along to Kinjal for our talk today.
Thank you very much, Vidaza. Okay, it's really a pleasure to be here and welcome everyone to the presentation. This is we're just kind of restarting community talks virtually, so I'm kind of excited to be sharing this with you. And I may have met some of you in person at the in-person Wellspring talk, so welcome. Today we'll be talking a bit about cancer genetics and epigenetics. Like Vidaza said, please feel free to type in your questions at any point of the during the session, and then we'll try to address them at the end as much as we can. So as Vidaza already said, I'm a volunteer at R.I.O.T, which is the Research Information Outreach Team. We have branches all across Canada, and we're basically a group of researchers who are affiliated with the Canadian Cancer Society, and we do community talks like this one. We also do high school talks, post blogs, and try to do community events in which we speak about the progress of cancer research. And so that's who we are, cancer researchers. Our goal is to share our knowledge of cancer and highlight current research. We're not licensed medical professionals, and we're not qualified to give any kind of medical advice. So for any specific questions medically would be best directed to a family doctor, an oncologist, but we're happy to answer any questions you have about cancer biology and cancer research. So getting back to what we're going to discuss today, we're going to talk about what cancer is at the molecular and the genetic level. Just get a little bit into the biology of cancer. And then we're going to explore the concept of epigenetics and talk a little bit about the epigenetic basis of cancer, which is sort of a hot topic in the field these days. And then a little bit of the behind the scenes of what we do in research labs. So this is actually an actual picture of one of my lab members looking at some cancer cells under the microscope. And that's a lot of what we do in the lab, just kind of studying these cells. And I'll be sharing a little bit of my research, if time permits, at the end of the session. But before we talk about cancer, we know that our understanding and study of cancer rests on what we know about normal cells. And so human tissue or human organs are composed of cells, which are the smallest unit of life. All cells perform basic functions such as respiration, which is to generate oxygen, and then power generation to sustain themselves, waste removal, and then of course cell division to maintain themselves. And this is on the right, you see a very basic representation of a cell. The basic cellular functions that I described in the previous slide are performed by these components called proteins. And in our cells, the DNA is actually what carries the instructions to make the proteins through a two-step process. So DNA is the genetic material of our cells and carries all the information from generation to generation. DNA gets converted into this intermediary, which is known as RNA, and then RNA ends up making the protein, and then proteins carry out all the functions of the cells. So you can imagine that every step is very important. It's very important to have intact DNA, and it's very important to have well-functioning proteins. So proteins carry out all these functions, all the basic ones that we discussed on the previous slide, but also specialized functions. So a melanocyte is a type of skin cell. And in the melanocyte, proteins tell the cell, make melanin. which is a specialized function. And melanin is actually made in order to protect our DNA and the skin cells from UV damage. So it carries out a purpose, but it also proteins also tell the cells divide or stop dividing, and this is all basic functions that happen in every cell. So cells rely on proteins to function properly, and they have a system of checks and balances to make sure everything is working smoothly. And so the specialized functions of the cells are sort of in part due to the nature of the cell. So for instance, melanocytes have specialized functions regarding the skin response or the skin functioning, but they respond to signals from the environment. And this is a very key feature of cells, is that the normal cells know when to stop dividing. And when needed, cells will undergo programmed cell death. And this is important because cells can often get damaged by the environment, or they could make mistakes in the way they carry out their functions. And when the mistakes are beyond repair, the cells do go through this programmed cell death. Of course, we know that cancer cells are different. So they do acquire abnormal characteristics. And so in the previous slide, we saw a normal skin cell, which has all these different components, the skin at the top, the hair coming through it, and the different layers, which carry blood vessels and nutrients and all of that good stuff. But in the picture over here, we see a abnormal skin cell, which has started to develop different stages of melanoma, different types of skin cancers, actually. So there's squamous cell carcinoma, basal cell carcinoma, and melanoma. And so cancer cells don't know when to stop dividing, and this is the biggest feature of cancer cells probably. Can't sense the environment, don't undergo cell death, they grow where they shouldn't grow, and the system of checks and balances basically fails in cancer cells. So cancer is a rapid and uncontrollable cell growth. And a good example of this is, you saw the picture of my friend looking at cancer cells in that dish, which had the red media, the cell culture media food for the cells. What we've noticed is that when we put normal cells from healthy tissue into that kind of dish, when they reach the edges of the dish, when they form a single layer, they stop growing because there isn't enough room for them to grow. But when we do the same with cancer cells, they start growing on top of each other, and they basically don't care that there isn't enough room. They just keep going, and that's a hallmark of cancer cells. And this is kind of what's created over decades of research, what's known as the hallmarks of cancer. So the abnormal characteristics that are uniform throughout different cancers are features such as evading growth suppressors, avoiding immune destruction. They induce blood vessel formation so that they can get a lot of oxygen and nutrients. They can kind of co-opt it from the regular healthy tissue. They resist cell death, of course, and the DNA also has many gene mutations, and that's what we're going to talk about in the subsequent few slides. And then this slide is a little bit packed, but I just wanted to highlight the sort of years and decades, maybe at this point centuries of research that has gone into understanding the normal genetic state of humans. And so there's a lot in there, but I'll just highlight a few different points. It was in the 1860s that the laws of inheritance were first described by an Austrian monk. And so parents passed certain traits on to their children. And so he realized that there must be something that's getting passed on generation to generation, but he didn't know what it was. And then it was only in the 1940s that DNA was identified as the genetic material. And then regulation, so gene regulation, and that's a topic that we're going to talk about a little bit more. That's what epigenetics is, was described only in 1960. The first human genome, as many of you will probably remember, was sequenced in 2000, which was a very, very big step forward for understanding the normal genetic state of humans. But only a few years later, a new project got launched, which was to map not just the genetic material of human cells, but the epigenetic material, which is a level of control above the DNA sequence. And we'll be talking about that as well. And now in 2020, we have thousands and thousands of genomes that have been sequenced, and we're trying to collect a very good sense of how different people have different genetic material and what can go wrong in cancer. So abnormal characteristics of cancer cells can be attributed to genetic changes. And so we talked about how DNA ultimately makes the proteins that carry out all the functions in cells. So if there were to be a mutation in the DNA, then you can imagine that that mutation or the effect of that mutation gets passed down through the RNA and into the protein, and that could change the way the protein works, or maybe not let it work at all, right? And so we have the DNA within each cell containing instructions to make proteins. And these actions include signaling the cell to divide or stop dividing. And so if something were to get wrong with the DNA, the proteins could either become hyperactivated or inactivated. And if this change conferred a growth advantage to the cell, this could lead to cancer. So identifying what genes are important will help us understand cancer better. So far, approximately 1% of all human genes have been causally implicated in cancer through mutations. But despite that, many cancers cannot be linked to a specific gene. So there's still a need to understand this process a little bit better, or to find another reason why cancers are sort of creating those abnormal functions. Oftentimes, cancer involves multiple gene mutations. And cancer can also sometimes be caused by mutations outside of genes. And this is sort of a very interesting feature that we discovered in 2000 when the genome was sequenced, that a very small percentage of the human genome is actually creating genes, or is actually genes. And genes are defined as the units of DNA that are going to eventually make a protein. Or write the instruction for a protein. And so many of you may be surprised to learn that less than 2% of DNA is actually a gene or genes. And so that's something we'll address a little bit later on. Scientists learn about mutations by sequencing both healthy and cancerous DNA and comparing them. And so I talked about how in 2000, the first human genome was fully sequenced, start to end. And since then, thousands of genomes, both healthy and cancerous, have been sequenced and compared, and a lot has been learned since then, and a lot more has to be learned as well. But as I mentioned, cancer could be beyond just genes. And so despite all that's known about cancer genes, many cancers are not linked with a specific gene. And in the previous slide, I had a picture of the BRCA1 and BRCA2 genes that are often mutated in cases of breast cancer. They only account for less than 5% of cases. And so there's the BRCA1 and 2 mutations here that kind of account for approximately 5 or 6% of breast cancer cases. And then there are other breast cancer gene mutations that might account for another 10% or so. But then we have this full, maybe 60 or 70% of the pie where there isn't any gene mutation necessarily attributed to that cancer. And so we don't really know what gene to link the formation of this cancer to. And so there's a lot of questions there about what's happening. But the answer might lie in the fact that genes are approximately 2% or maybe less of our DNA. And so there's a lot more DNA in our cells that doesn't make proteins. So what is it doing, right? And that's kind of where the term epigenetics comes in. Epigenetics allows the exact same DNA in every cell to create different kinds of tissues. And so we all come from embryonic stem cells or fertilized eggs from our mother. And these cells, which were initially a single cell, start to form different kinds of cells. But you can imagine that that single cell that eventually forms all these different cell types, liver or hair, brain or skin, had the exact same DNA. So every single cell in our body has the exact same DNA. We know that about 2% is what we call genes, and the other 98% used to be called junk DNA, or the dark matter of the genome. But what turns out is that 98% helps control the 2% in a way that it can perform different functions and create all these variety of cells that make up the human body. And so here's kind of an example of what I'm trying to illustrate. genes are scattered throughout our DNA, and there aren't a lot of them compared to how much DNA we have. So this kind of looks like just a bunch of gibberish. Let's imagine that this is our DNA, which is just made up of random letters strung together. But then we start to see a pattern. And so this is the code for making skin cells. And so these are the few genes that are present in skin cells that are active and are helping making proteins for skin cells. This may not be too clear. If you have any questions about it, please feel free to type it in, and I'd be happy to address it. But what I'm trying to say is that the genes in our cells are not active all the time. There are some specific genes that are very specialized, let's say for skin cells, and so they would only be active in the skin cells. And then we would have a completely different set of genes active in brain cells. So there are some genes that are shared. We know that there are proteins that carry out universal functions like energy and respiration, but then there are completely specialized functions, and those specialized functions come from specialized proteins that are instructed by specialized genes. And what controls these different sets of genes to be on and off in the different types of cells is what we call the epigenetic code. So you could say that if our genetics or our genetic material is a book that needs to be read top to bottom or start to end, epigenetics form the bookmarks that have different sets of bookmarks for different types of cells that allow the genetic material to be read and interpreted correctly for that specific context. And so you can imagine what would happen if something went wrong with the epigenetic signals, right? And so what epigenetic signals do is that they sort of hang around very close to genes or perhaps further away. They control genes from a different location than the gene. They can signal the gene very much like a traffic signal to turn it off. So if this gene is very specific for a skin function, then this gene would be turned off in the brain cell. Conversely, in the brain cell, another gene that's important for a brain function, let's say making a neuron or a nerve cell, that gene would be turned on, and so there would be a different epigenetic signal at this gene instructing that gene to be turned on. Epigenetic signals can be quite sophisticated, and so sometimes they can even keep the genes paused until they're ready to be turned on, and often what allows them to be turned on or off is a signal from the environment. And so epigenetic signals can control gene function without changing the DNA sequence. So they decorate the DNA sequence itself. As you see here by these little, what look like little flags, they're actually chemical signals or little chemical modifications tacked onto the DNA, like little traffic signals, and they each represent something. They each represent a green light or a yellow light or a red light. And so that's how they ultimately control how genes are acting in different ways and in different cells. Okay. So let's come back to this analogy where different genes are active in different types of cells. You can imagine that if this epigenetic element that's right beside the gene responsible for brain function got changed in some way, then that gene wouldn't be able to work properly, and that could control or disrupt the gene activity in that cell. So an altered epigenetic code can actually end up promoting the growth of a cancer in that way. And so we've got a gene here, and then surrounding the gene are these epigenetic signals, or these chemical signals that they act as tags or as traffic signals to control what's going on with the gene. So in this case, a gene that's typically meant to be off in a certain cell could be epigenetically hyperactivated and turned on. Conversely, a gene that's meant to prevent tumors from forming or prevent cancers from growing could be epigenetically suppressed, where the silencing tags get tacked onto that gene, and that gene is no longer allowed to act and prevent the growth of a cancer. So these are some ways in which epigenetics, or an altered epigenetic code, can promote the growth of a cancer. And then finally, the activity of genes which are involved in responding to cancer treatment could also get altered by epigenetics. Another way that epigenetics can sort of interfere with the functioning of a gene is physically. And so initially we talked about chemical changes what prevent the gene from being turned on or off. But the structure of DNA is actually very, very important in determining gene function. Many of you may be surprised to hear that we, in every single cell of our body, we each carry 2 to 3 meters of DNA. If you stretched out the DNA in every cell, that would be, just well taller than I am. I'm about like 5'3". And so the way that DNA gets incorporated into such a tiny little cell is through intense folding. It gets folded and refolded in different ways in order to get packed in. But when genes need to be functioning to make proteins, the bit of DNA that has that gene unfolds a little bit and then allows the gene to do its thing and then refolds again. And so epigenetics can actually control how the DNA is folded. And so it may prevent an important gene from being unfolded, or it may fold up a tumor suppressing gene and make it less exposed to carry out its function. And so this way, aberrant epigenetics can cause cancer promoting genes or genes involved in cancer to be more or less exposed than they should be and affect their functioning that way. And so we're coming back now to the hallmarks of cancer, but this time we're attributing these changes or these characteristics not to gene mutations necessarily, but to an altered epigenetic code in those cells. And so changed epigenetics can cause any of these features and promote the cell to have cancer. I should say that most cancer cells have genetic mutations as well. And so cancer cell usually has changed DNA and then also an additional layer of changed epigenetics. So both changed genetics and changed epigenetics to create a perfect storm in which the cell starts to divide uncontrollably. So there are several drugs that target epigenetic features that have entered into clinical trials for cancer treatment. And I'll take you really quickly through the way that this sort of happens. It begins in labs, such as the lab that I work in studies known as preclinical studies. And so you could have a screen for different drugs that we try out on cancer cells to see, and these cells are in the dish, to see basically which drugs affect the growth of cancer cells or other characteristics of cancer cells, such as the capacity to invade or metastasize. And so we perform screens like that, or we perform basic biology to look for biologically what would be a meaningful target to be drugged. Then we perform a number of what's called in vitro, because we're still doing it in cells in a dish, large-scale studies where we sequence the cells after they've been treated with the drug, and we perform a large number of tests to see what effect the drug is having on these cells. This is called in vitro. Then we move into something that's called in vivo preclinical drugs, where different models of disease are used. Often it's mice, because mice are pretty similar to human beings in the way that they respond to drugs and in the way that their biology occurs. These drugs are tested later in mice to see how well the mice tolerate the drugs and if it's effective at treating the cancer that is arising in the mice. And then when it passes these different stages of preclinical studies, it moves into clinical studies in different phases. And so these are the different drugs that have gone through all of these different phases of clinical trial studies. And I'll give you one case example. It's called Vidaza. And so what Vidaza does is that it prevents a chemical modification or an epigenetic signal from being tacked onto the DNA. And what that epigenetic signal does is that it prevents a gene from making a protein. And so Vidaza prevents that signal from being added on, which means that it breaks that, that repression. And so here's an example of what could be happening. This is the chemical signal that Vidaza actually blocks, but if there's no Vidaza, then this chemical signal is able to suppress genes that might be preventing tumor growth. So you do have a tumor. However, when you add Vidaza, it kind of prevents those chemical signals or those epigenetic signals from being added on. This allows the gene to actually make the protein and carry out its role as a tumor suppressor, and therefore you can have a prevention of the tumor forming. This is a very sort of representational example of how it might work. It doesn't, of course, work in every type of cancer, but it does work in some. And an example of that is acute myeloid leukemia. And so there was a clinical trial done with Vidaza that showed that patients treated with Vidaza that had AML were able to have a better outcome than patients that weren't treated with this drug. And we're still good for time, so I'm going to maybe just spend a few minutes on the kind of research that I've been doing, and then we'll open up the platform for questions and just kind of a discussion, which I would really enjoy. And so, like Vineta said, I work on a cancer called Medulloblastoma. It's a type of brain cancer that occurs primarily in very young children. And the tumor, like most tumors, is very heterogeneous. So there are different types of cells, and they have different capacities for growth. And so a type of cell in that tumor, it's called brain tumor stem cells. And these stem cells are very, very resistant to traditional chemotherapy, which means that chemotherapy cannot kill those cells. And once chemotherapy is done, the cells are actually able to repopulate a tumor resembling the original tumor in the first place. And so one of my research goals is to identify the epigenetic differences between the different types of cells in the tumor and try to basically eliminate the stem cells along with the rest of the cells. And so the way I did that, the method that I used was to do an epigenetic profiling of the stem cells versus the rest of the tumor to find out these differences. And what I noticed, as you can see from this plot here, which is actually my unpublished data, is that I created epigenetic scores at all the different genes in the cell. There's about 20 or 25,000 genes in every cell, not all active, as we now know, but there was huge differences between the brain tumor stem cells and the rest of the tumor, which is actually less capable of dividing and creating the rest of the tumor. So what I wanted to know is not only how are they different from each other, but can this difference be targeted? And so I identified a gene that's very, very different between the two in terms of its epigenetic score. And I tried to target it specifically in the stem cells. And so, I'm sorry, the slide hasn't rendered very well, but here's just a representation again of the medulloblastoma tumor with the different types of cells in it. On one side, we have the traditional therapy that eliminates most of the tumor, but not the stem cells. So what I decided to do is target that one gene that looks to be epigenetically different in the stem cells. I applied a novel therapy eliminating the effect of that gene. And the goal is that you can get rid of the stem cells. The rest of the cells will sort of die off in time or not be able to sort of propagate themselves as well without the stem cells, and ultimately we're left with no tumor. So I actually used CRISPR technology, which some of you may be very have heard of, which sort of is able to make changes in the DNA. And so using CRISPRs, I took the DNA, which was without any mutation in that gene, and made the gene into a mutant. And as we learned in previous slides, when you make a mutant gene, then the protein is not able to work very well. And so I effectively kind of canceled out that protein in those tumor cells. And then I implanted these cells, both unmodified protein as well as the modified, into mice, and then measured the growth of tumors. And what I noticed is that in the non-edited cells, you can see that tumors rapidly start to form and grow in these mice, whereas in the CRISPR-edited cells, which are in purple, there was an inability of the cells to form a tumor in those mice. And so by just eliminating the one gene, I was able to see this very drastic difference in the growth of tumors. And so this is something that I'm now developing in different ways and trying to understand and study in different ways as a potential additional therapy to supplement traditional chemotherapy in medulloblastoma. And so I'll stop with that. I'd love to take questions. We have about 25 minutes for discussion, which is plenty of time. So I'm hoping that we can have some chats and a lot of you can ask in some questions. But I just want to put up here our website. It's torontoriot.com, where we write blogs about cutting edge and new and interesting research in the field of cancer. And we also have a Facebook page and a Twitter account. So feel free to follow us and share. And with that, I'd be happy to take any questions.
So thank you so much, Kinjal, for that presentation. It was super informative. And so now we're going to move on to the Q&A segment. So our first question, how does Herceptin work on the HER2 positive protein?
Yeah, that's a great question. So in the Herceptin, so breast cancer has multiple subtypes, as you know, and one of them is called the HER2 positive breast cancer. And the reason it's called that is because this gene called HER2, it's involved in pathways of cell growth. It becomes mutated in a way that there are multiple copies of HER2 present in the DNA. So it gets amplified. And because it gets amplified, a ton more of HER2 protein is getting made. And HER2 protein helps the cell grow. So the cell starts becoming addicted to the growth that's pushed or spurred by excessive HER2 protein. What Herceptin does, is block the protein from acting. And so a ton of HER2 is being made that is helping the cell grow, and Herceptin is just physically blocking HER2. And so the cell, which has become very dependent on HER2 for dividing and kind of propagating itself, is unable to do so. And to date, Herceptin is actually one of the best and most effective therapies that we found in cancer.
Perfect.
I hope that addressed your question.
Yeah, that was great. Thank you so much. So our next question, do epigenetic clinical trials work on BRCA1 ovarian cancer?
Yeah, that's a good question. So epigenetic trials are epigenetic research is being conducted in every single type of cancer you can imagine. So the short answer is that it's being researched. I don't know yet if there are any drugs targeting epigenetic factors in the cancer that you asked, ovarian, I believe. So I don't know, but there is an excellent website, and it's in the resource sheet that I sent Vineeta, and I'm sure she would be circulating to all of you. It's called clinicaltrials.gov. And I would sort of just look at the trials that are going on in that cancer, and they do follow up with information about every drug that's in trial, and would let you know if there was an epigenetic drug. But there's also a difficulty with epigenetic drugs because, as you know, epigenetics is a part and parcel of regular cell functioning and regular development of the cell from embryonic stage until where we are now, and epigenetics need to keep functioning for us to be healthy as well. And so epigenetic drugs do have a lot of off-target and widespread effects in our genes. If it's able to balance out with a very negative cancer-causing gene that gets kind of stopped, then the epigenetic drug would be effective. But for many cancers that are purely driven by genetic mutations, epigenetic drugs are actually not very effective. And so in the cases of BRCA mutations and things like that, it's pretty much a one-to-one cause and effect, where we know that the cancer cell is being spurred by changes in the BRCA gene, which is a DNA repair gene. And so in those cases, perhaps epigenetic therapy would be less effective.
Perfect. Thank you so much for that. And so our next question, I think this one is a broader one.
Okay.
How do you change the genes of all the cells in the body?
Wow, that's a broad question.
That's a big open question.
That's a big question, and you're probably asking something that scientists are thinking about all the time. So great question. There's so many ways to answer this, right? And so how do you change all the genes in the body? There are different ways to change genes, and we've talked about epigenetics for some time. And so it is sort of a level of regulation that we can't really control, but we can just observe as researchers. And there are different epigenetic proteins that sort of recognize different features of the DNA, and they have their, let's say, target genes. They go to those genes and then they make changes on those genes as needed. And epigenetic factors are also influenced by the environment. So let's say we're in a situation where there's less oxygen, certain responsive factors to the level of oxygen, so they're able to sense the level of oxygen in and around us, in and around our cells, sort of go to genes that would help kind of push up the metabolism of oxygen or the use of oxygen in our cells. So it's very, very dependent on the environment, and it's very difficult for us to control. So that's one way in which epigenetics sort of starts playing around with the different genes in the cell. The other way that genes can change is through mutations. And so in the example in my research that I showed, I actually went in and made a mutation in a gene. But it's very, very difficult to do this in cells, because I was able to do it in a dish where I can sort of like manipulate cells. In human beings, it's much more difficult. And the regular functioning of the cells, it's also much more difficult. So to answer your question, you know, cells have their own way of working, and that's how genes are very dynamic. But it's something that we are still trying to understand.
Perfect. Thank you for that. And so our next question, how do the triple negative cancer cells function?
Oh, well, yeah, that's a good question. Of all the breast cancers, we know the least about triple negative cancers. We know that they do have mutations in BRCA1 or 2, which is inherited. It may also have other mutations that we don't really know yet, or it may have a collection of mutations, or it may have epigenetic changes. But the way that they divide and kind of propagate is through breaking down the checks and balances in the cell. And so BRCA1 and 2 are involved in DNA repair. And basically, when there are, and so I should say that mutations are acquired in every cell all the time, but they're corrected. They're corrected by the machinery of the cell itself. And so the cell catches so proteins that are responsible specifically for this, like BRCA, catch the mistake, they get other proteins to come correct it, and if it can't be corrected, then they give the signal for the cell to undergo programmed cell death. But when BRCA is mutated, the cell ignores that. There's no one to say, hey, you can't divide, you have a mutation, you're gonna make mistakes, and you're gonna divide uncontrollably, you're kind of out of control. So BRCA is the one that does that. That's kind of the check and balance that gets missed. And that's how triple negative cancers sort of propagate uncontrollably. And that's kind of how it works for them. But I should say there are many unanswered questions in that as well.
Thank you for that. And so our next question, I think, is a little bit more of a specific individualized question. So I'm going to try to make it a little bit more general. But there are companies that test for more genetic mutations than are covered by the genetic tests covered by OHIP. Like, for example, Logica targets 505 genes covering oncogenes, fusion genes, genes susceptible to copy number variation, and tumor suppressors. So for individuals with maybe a rare form of cancer, like for example, maybe a rare breast cancer, would these tests be something that would be useful?
Just in terms of like individual cases, it's very difficult to say. But my general response to that is that a lot of these genetic tests done by OHIP or other places do cover a very limited range of genes, but the genes that are covered, as I mentioned, about 1% of genes have been implicated in cancer, which is about 200 genes. So 500 is a lot more than that. The reason it's very difficult to find mutations sometimes in rare cancers is because the mutations are occurring not at these commonly mutated genes, but perhaps at the junk DNA that's controlling genes. These mutations are very, very, very difficult to find. My answer would be that, you know, your oncologist would probably know better than anyone, but it would probably be difficult to detect mutations in such a cancer, just because we don't know the broad mechanism by which what's going wrong. And this is definitely an area of research. And I would also say that these non-gene mutations or these non-gene epigenetic processes that are happening to converge on pathways that promote cancer are being studied and ultimately will move into the clinic as well.
Thank you for that. And so our next question, what impact does a blood transfusion change the genetics or epigenetics in our body?
Oh, wow. That's a great question. To be honest, I don't really know the answer to that. It wouldn't change the genetics because we're all born with the same DNA in all of our cells, and having a different type of cell come in with a slightly different genetic makeup is not going to change the existing genetic settings that we already have. In terms of epigenetics, it is a really good question, and the reason is because we know that epigenetics can be influenced by our environment, and so blood cells mingling with each other that are slightly different could trigger some kind of epigenetic. The main way in which probably there would be a change would be within the immune system, which is influenced by epigenetics also, but also influenced by the way that proteins end up presenting themselves to the immune cells. And so long answer short, like I don't really know, but my guess is that it wouldn't make too many changes, and it certainly wouldn't affect mutations, for instance, or genetics or epigenetics that promote cancer.
Oh, sorry. I thought I had unmuted myself. I was going to say
okay.
thank you for that. So it looks like our next question is a question regarding R.I.O.T's research. And so the question is, do you examine other processes that exist in cancer treatment that is outside of pharmaceuticals?
Yes. And so, like I mentioned, R.I.O.T is a team of volunteers, but we all work in labs in different capacities, either as students or postdocs, things like that. So we each work in very different areas of research, but specifically in my research, we are examining, a lot of different, I think maybe your question is regarding the screens. The answer is yes. So we look at all available drugs, and that's the easiest place to start because they're already FDA or Health Canada approved. And so repurposing them for other cancers sort of cuts the clinical trial research time by a lot. So from where we stand now, it takes about 15 or 20 years and like over $1 billion to get a drug from basic research into the market. And we can definitely cut that by over half with readily available or on the market drugs. But like the research that I did, for instance, did not start with a drug. It started with a biological question where I kind of went blind into it, looked for differences, found the most different gene, and then used a way to target it. And now it can be a good candidate for a drug. So we do look at processes, including epigenetic processes that don't already exist as drugs. And then we try to find a company that would either be willing to make the drug or work with us in order to like sort of get that drug going for the next stage of preclinical research. And then finally, we do other kinds of screens. And an example of this is that because I study a childhood brain cancer, we're asking the question, is something happening while the mom is pregnant to affect the way that the child's brain is developing, or kind of maybe predisposing them to get cancer? We don't really know, right? Like it's all open questions. So we do screens of things that might be possibly toxic to a developing fetus. And so there are many, many ways of approaching these questions, and we try to address as many as we can across the world.
Thank you for that. And so our next question, would you be able to speak to what causes the BRCA gene mutations?
Oh, well, yeah, it's a good question. Ultimately, most mutations, there are a few different ways in which mutations are created. One of them is through just a common error, just an accident in the cell. Because when cells divide, they actually have to double their DNA, split it apart, and then form two cells. And that's how a cell divides. So to double the DNA, it has to kind of read the DNA and then make an exact copy of it. So, I mean, I make a lot of mistakes reading and writing things, just copying one to the other, and so does the cell. So when it makes mistakes, those mistakes often get corrected. But sometimes it makes a mistake in a very key gene, like BRCA1. The mistake in that gene is providing a growth advantage to the cell. And because of that, it retains the mutation, because the cell has become so much faster and better at dividing that it kind of outgrows all the normal cells that are dividing at a very contained pace. And so accidents in cell division or in DNA, what's called DNA replication, can result in mutations if those mutations provide a growth advantage to the cells. So you can imagine that that mutation happened in one person, in one cell by complete accident, but it kept growing in a way that it sort of overtook. And then that mutation got passed on from mother to daughter, father to son, however it happens, right? And because we inherit equal genetic material from both parents. And so if the mutation is strong enough that it can change the way the cell grows, it gets passed on from generation to generation. The other way that mutations can happen is through external mutagens like ultraviolet radiation or ultraviolet rays, which can actually cause mutations in our cells too, although those cells would most likely be the skin cells, which are exposed constantly to that type of UV rays. And so mutations can happen in a number of ways, but then they'll stick around if they benefit the cell in any way. So now breast cancer or BRCA1 and 2 gene mutations have become fixed. They have the ability to be passed on from parent to child. I hope that answers your question, but it's not always the way mutations happen. Anyone can get a mutation at any time. Yeah, and sometimes they go uncorrected.
Thank you for that. And so we have kind of a two-part question here.
Okay.
So one, if you would be able to speak about any recent research you're aware of in regards to breast cancer specifically. And the second would be, how does letrozole fit into breast cancer treatment?
Okay, I'm not really, unfortunately, a clinical scientist, so I'm not aware of all of the drugs targeting different cancers, but I will tell you a little bit about breast cancer research that I was involved in during my PhD. It was actually a very epigenetic related project, and what we noticed is that in estrogen receptor or hormone receptor positive breast cancers, which are about 65 to 70% of all breast cancer cases, estrogen receptor is a gene that's also involved in growth pathways of the cell. And like HER2, the cell becomes quite dependent on estrogen receptor to grow, and it gets targeted by the drug. So we saw a large increase in the estrogen receptor proteins in the cell, but we didn't find any mutations in estrogen receptor. So that was the problem that I wanted to solve. Long story short, I should say, full five and a half year PhD summarized in this like next 2 sentences. What we found is that the junk DNA, or the regions surrounding the estrogen receptor gene, were the ones that had the mutations. And so we were able to show that if we got rid of those mutations themselves, or we got rid of those regions, we were able to bring down the levels of estrogen receptor and actually stop the cells from growing like a cancer does. And so epigenetics is a big part of the growth of breast cancers, specifically estrogen receptor positive breast cancer. And the next step, of course, is to find a way to sort of target those regions specifically and prevent them from acting on estrogen receptor. We do have a breast cancer talk coming up in a few weeks as well, where we'll have even more insights into new breast cancer research that you're welcome to attend. And then speaking of letrozole, I'm afraid I don't know enough about it to comment.
Mm-hmm. Okay, thank you for that. And so our next question, I'm not sure if you would be familiar with this, but do you know when CRISPR will be used more broadly?
Yeah, that's a really good question, and definitely a big debate, and not a debate, but a discussion in the field of among scientists like myself. Like I mentioned, I use CRISPR in my research, and it's a really great tool to understand how genes work, because we use CRISPR to basically inactivate the gene, and then we see what changed. So CRISPR is an excellent tool in research. It's not really ready to be used clinically, because it's we need to rely on CRISPR being highly specific to work. And right now, CRISPR still has some problems, like it would make the mutation that we wanted, but it would make other mutations too, which are unwanted. And so I would say that it's really tough to predict, but I think that CRISPR technology keeps getting more honed. Of course, there is a whole ethical debate that surrounds it as well, which is that once we can start using CRISPR in human embryos, where's the line, right? And the other issue is that we've already kind of discussed this in the previous slides, is that often cancer is not caused by one gene, and I would say it's almost never caused by a single gene changing. And so changing back the mutation in that one gene may not actually stop the tide. So what we have to do as researchers is to understand the complexity of what causes changes in a cell to form a cancer, and then try to maybe find the root of that, which may not be in one gene, but maybe in a process or in a pathway or in a group of genes. And we can try to find the mechanism and then use drugs to target those mechanisms. The other thing is that it's impossible to use CRISPR in a fully grown human, because CRISPR can only work in a single cell. So if the CRISPR change was made in a human embryo, potentially, this is not yet, it's not allowed to be done because of ethical and scientific restrictions. But you could make the change in the embryo, implant the embryo would form with the genetic change constant in all the cells of its body, because they all came from one cell. But if I wanted to make a change in some cells in my arm, I wouldn't be able to do it, because I'd have to take the cells out, put them in the dish, do the CRISPR, and then put them back, and then only those cells would be changed. So it's still a developing and evolving technology, and it's at least 5 to 10 years before it can be used for any kind of clinical impact.
Mm-hmm. Thank you for that. And so our next question, is it possible that mutations disappear over generations?
Wow, that's a great question. I love that question. The answer is yes. So mutations are constantly forming and disappearing in our cells, in our bodies. The mutations that stick either have some kind of a growth advantage in the cell or they have no change at all. So it doesn't matter to the cell whether the mutation is there or not. I sometimes speak about cells as if they're their own people, you know, like they have their own minds, but just think about if there was, if you were walking down the street and you had some kind of thorn in your foot, you would remove it because it's causing you pain. But if you happen to walk on some kind of magic thorn that didn't cause you any pain but helped you walk faster, you wouldn't get rid of it. Or if something happened and it didn't cause you pain, it didn't change anything, you're like, it doesn't matter to me. So that's how mutations are. If they cause pain, the cell gets rid of them, and over time they will disappear. But if it actually gives the cell some kind of growth advantage, or it doesn't change anything, then those mutations tend to stick around. And sometimes the mutations that give a growth advantage are the same mutations that cause cancer ultimately. And so mutations are very dynamic and they can come and go. The ones that stay are the ones that change the cell positively.
Okay, perfect. So thank you so much for answering all of those questions and for such a wonderful talk. I think we're running close on time here, so I'm going to wrap up the question and answer segment. So I apologize if we weren't able to get to your question, but Kinjal, thank you so much for answering all of the questions, and I will be sending out the resource sheet later today.
Perfect.
And I know we had another question about the breast cancer talk that you mentioned. So that one, I believe it is on just
24th.
Yes, the 24th,
yeah.
1:30 to 2:30. So if you are interested in that, feel free to register for that on our website as well. And I think Kinjal is also presenting another talk relating to the history of cancer and progress in cancer research. And that one is on July 15th. So we have a lot of Be Well talks, so feel free to just go on the website and check out whatever interests you and sign up for those ones. And so I think with that, we'll wrap it up for today. And I just wanted to thank you, Kinjal, once again, and everyone who was able to join us today.
Yes, thank you very much for attending and for all your great questions. And hopefully you'll be encouraged to attend some of our other sessions as we all move and adapt to this new technology. So thanks, everyone.
Thank you so much and have a great day, everyone. Thank you, Kinjal.